Hello, everyone, and welcome to today's live broadcast, Phenotypic Profiling for Improving Drug Discovery Productivity, presented by Dr. Ellen Berg. She is the Chief Scientific Officer of Translational Biology at Eurofins DiscoverX. I am Marjorie Torres of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during this presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom or center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Ellen Berg. I will now turn the presentation over to her.
Thank you. It's great to be here. Happy to be speaking to you about my thoughts about how we can improve drug discovery productivity. Just one note about conflicts of interest. I am an employee of Eurofins DiscoverX, and I will be mentioning a little bit about a couple of our platform technologies. But most of this presentation is certainly my opinions and not necessarily the opinions of Eurofins DiscoverX. So if we start here, what I'm going to talk to you about today is the crisis in pharmaceutical productivity, talk a little bit about where we are with the current drug discovery paradigm and what I see as weaknesses in the paradigm, and then present to you some of my thoughts about solutions that are coming from innovations in new technologies, better models of disease, in particular in vitro models, and advances coming from combining in vitro and in silico methods. So everyone is probably familiar with the pharmaceutical productivity crisis. Certainly we know that our drug costs are very high. And one of the reasons driving this is the fact that more than 90% of drugs that go into clinical trials fail. And they mostly fail for efficacy or safety reasons. So obviously contributing to high drug costs and problems for both patients as well as companies. And, despite all of our investments in omics technologies, genomics, proteomics, et cetera, and high throughput methods, our success rates have not been improving. They're stagnating. And keep in mind that we completed the sequencing of the human genome over 15 years ago. So we anticipated doing a better job in 2018 than we are with success rates in the clinic. So if we take a look at the current drug discovery paradigm, the typical target-based drug discovery paradigm, where do I see the weaknesses? So typically targets come from genomics. Compounds are screened against those targets, and medicinal chemistry is applied to optimize leads, and they go into preclinical animal testing and then subsequently human testing. So where I see the weaknesses in this paradigm are our reliance on genomics-derived targets and also our dependence on animal testing for making decisions and determining whether or not we're going to go forward with a program and go into the clinic. So the way I see the problem with genomics is that we are suffering from genomics addiction. It's easy to get a lot of data. There's lots of cool tools to visualize the data, giving us what I believe is an illusion of knowledge. My issues with genomics is that typically the data are not in context in terms of a cell type, a condition, a patient, organ, disease stage, etc. And also, gene expression is inside the cell, so it's also too far from the action, the outcome of what's going to happen in a patient, which is at the level of the organ and organism. So I think of genomics really as being machine language, whereas biology and clinical outcomes are object-oriented programming. And so it's very hard to predict from genomic information how the program is going to work. And that's one of the key weaknesses in our current paradigm. The second weakness I see is dependence on animal models. So what's the problem with animal models? Aren't they great models of disease? Well, the problem is our favorite models, usually typically rodents, don't live very long compared to us, 2 years versus 70 years. They're very small compared to people. And the environments that they are housed in are very clean, very standardized, and do not contain a lot of the exposures that people have in terms of from viruses and bacteria and saliva from our pets and our children. So we are exposed to quite a different environment than animals that are used in pharmaceutical research. This has a huge impact on some very important areas of biology that are studied for drug discovery. In oncology, the DNA repair mechanisms. If an organism only has to live 2 years, it doesn't need much in terms of repairing DNA because it's not going to be around long enough for those collected mutations to cause any harm. Whereas people live for so many years, they have to have a lot of different mechanisms to take care of mutation rates. Control of blood flow, and a very tiny organism isn't subject to gravity as much as we are. The size of our blood vessels of humans is much larger than in animals, and actually the distance of a tissue cell, the average distance of your tissue cells from the closest blood vessel, is much greater in people than it is in rodents, and particular mice. So control of blood flow and hemostasis is quite different between humans and mice, of course contributing to cardiovascular, the ability to predict how a cardiovascular drug is going to work in a person. And most of all, immune system status. Mice and rodents, their immune systems are relatively naive. They haven't been exposed to many stressors, bacteria and viruses, whereas people's adult human immune systems are quite differentiated and activated and educated. So they're quite different. Our response to and diseases such as autoimmune, asthma, allergy, the mechanisms are quite different in people. So these are some of the reasons why, depending on animal models to determine whether or not a drug is going to work into a person, doesn't work so well. And those are just a few of the areas where the differences in animals and people makes it problematic to discover drugs that are going to work. What we do need is a mechanism or solution to incorporate the context and complexity of human biology into our drug discovery paradigm. We need to remember that, or take into account, build in the fact that biological systems are modular and hierarchical. So no matter how much we understand about how a drug interacts with a protein target at the molecular level, that does not predict how that target will work in a pathway, how pathways might interact and combine together in a cell, how cells interact with tissues, and so on. And so we have to incorporate higher levels of organization in our solutions. So the way we do this is to think about building better models of disease that are human relevant, that do incorporate these issues, and be able to, by combining in silico and in vitro approaches, work towards improving prediction of drug action in people. I will say that in silico models alone aren't the solution. Many in silico models, predictive models, rely on published literature, and we have to recognize that there's a known problem with reproducibility in the scientific literature. There's a lot of poor quality publications arising from misaligned incentives and explosion in predatory journals. And there's also a persistent bias coming from the fact that people are much more likely to publish positive studies than negative studies, not to mention statistical cherry-picking issues that cause problems with taking in only published literature. In addition, there is a often misapplication of predictive models. There are many models that have been published that are out there where there's really been insufficient data going into the models, or overfitting, so that when you introduce new test data, they do not work very well. There's a lot of global models that tend to be trivial because particularly most models of drug-induced liver injury, yes, they work for a percentage of drugs and compounds, but it's not at the level to be useful. Useful model performance in this area needs to be 95 plus 99% accurate to be useful, because often the events that we're predicting are rare, and very high performance is needed. And typically you can only get that level of performance when you go to local models that are very specific. I'll talk a little bit more about that later. So what do we, if we think about our task here, how do we do a better job of predicting drug action in people? There is a role certainly a role for better in vitro models of disease, because we can use human cells so that they would be human relevant. There have been a lot of advances in cell culture innovations for being able to culture human cell types, that also techniques that allow us to run assays in very high throughput with a quality mindset, so very reproducible and robust. In terms of their physiological relevance, we advocate following the Pfizer's phenotypic rule of 3, where your most useful assay will employ the highest physiologically relevant cell type that you can work with, using physiologically relevant culture conditions, and selecting assay endpoints that are proximal, close to clinical endpoints. So the most physiologically relevant cell types there are to use in culture remain human primary cells. These are cultured, taken out of normal or patients, and immediately cultured and used in assays. They're not extensively cultured, unlike iPSC cells, induced pluripotent stem cells, or other stem cell-derived cell types, which must be cultured for sometimes weeks and months to differentiate these cells into their adult counterparts. Unlike cell lines, primary cells are not mutated and not long-lived. And of course, co-cultures of different cell types, which could represent tissues, would be even more relevant. through the use of human cells, obviously the results can be directly compared to human clinical. There's no concern about species differences in using human primary cells. In terms of what to use for the most relevant endpoints to measure in these assays, validated clinical biomarkers are the best. They're disease-relevant proteins or metabolites. They respond, they're associated with clinical disease and or responses to drugs. In particular, more useful proteins would be cell surface or secreted proteins because, or metabolites, because these are the most proximal to a clinical endpoint. They mediate communication between cells and tissues so that they're at a higher level of the hierarchical organization and serve as a natural bridge between in vitro and in vivo. And the third aspect here would be to appreciate that there's a lot of biology to cover, and one would do that through developing multiple assays that would tap into different aspects of tissues or cell types or disease settings. And this represents here a standard phenotypic assay panel built with human primary cells that's commercially offered through Eurofins DiscoverX that contains assay systems that cover vascular biology, respiratory biology, fibrosis, and immune and inflammatory responses. And so by using a panel of assays, one can cover a lot of a diverse range of biology. And the other important thing to do to have a system that can predict human outcomes is to test it. So by developing a large reference database and testing many, many drugs, experimental chemicals, compounds, tool compounds, validating these assay systems for their relevance, testing their relevance for clinical biology. So this is a way to develop a tool or a system that can help us identify those compounds that might do better in human clinical trials. The way that we have to validate these systems then is by applying data mining, right? So we, for example, here is a set of data across this particular panel of 12 systems shown in the top now. We're showing here a list of several different drugs along the y-axis. The x-axis represents the clinical biomarkers that are measured in each one of these assays. So in each system, on order of 10 to 15 clinical biomarkers is measured. And what we can do then is by taking a set of drugs, 1, 2, and 3 drugs that are, and we can look across at these biomarker activities and find common activities that are shared by drugs. For example, these 3 drugs share, even though they are against different targets and used for different indications, they share an adverse effect, and we can see that they share an activity. They all increase the level of a protein biomarker in one of the systems. That's shown by the blue indicates the protein biomarker level has increased, and the orange shows you where protein biomarkers are decreased. And even though their overall patterns are quite different between these 3 drugs, they do share this one activity in this assay, suggesting there's a potential hypothesis that this could be related to the side effect, because that's the one aspect that they share. And so in this way, we can use these large data sets to search for correlations between biomarker activities and clinical effects. So I'm going to share with you 2 case studies, both having to do with cardiovascular safety, which is a long interest of mine. The first case and how we've applied these these systems to validate assays for their application in predicting human clinical outcomes. The first case study has to do with cardiovascular toxicity of oncology drugs, in particular related to trametinib, which is a MEK kinase inhibitor that's approved
I can help you search the web
for
here an adverse effect, and we can see that they share an activity. They all increase the
and we can MEK kinase. And the second case study that I'm going to present has to do with thrombosis-related side effects, deep vein thrombosis and pulmonary embolism. So let's begin with the first case study. Trametinib is an allosteric MEK inhibitor, which was approved for the treatment of metastatic melanoma. Like most oncology drugs, it has side effects. These include rash, diarrhea, and cardiotoxicity that I mentioned. So what we had identified with this particular drug is a biomarker signature in this assay panel that was associated with trametinib and other drugs that are associated with, that have increased risk of cardiovascular toxicity. And this was increased levels, cell surface levels of a protein called Serum Amyloid A in a human primary cell-based assay of vascular inflammation, coronary artery smooth muscle cells, and trametinib increases the level of serum amyloid A, as did a couple of other drugs that were also known to have cardiovascular toxicities. And what was particularly interesting is serum amyloid A is it's an acute phase response protein, but it's actually a known clinical biomarker for vascular inflammation and a risk factor itself for cardiovascular events. So this was very encouraging, suggesting the association might be, could certainly potentially have clinical relevance. But we went to confirm this. We went forward with studying how this particular protein was regulated in this system, and to understand the positive and negative regulators of this protein, serum amyloid A, in our in vitro model. And so what we do is we take the profile, so the activity signature increased serum amyloid A, and run it through a reference database of about 3,800 drugs that had been profiled in this assay system, and identified all of those drugs that also shared this activity of increasing serum amyloid A. And I think it was 40 or 50 drugs. But what was interesting, and experimental tool compounds, what was interesting is that they fell into some very specific target classes and mechanism classes. MEK inhibitors, like trametinib, glucocorticoid receptor agonists, these are glucocorticoids, HDAC inhibitors, IL-6 pathway agonists, nicotine, and other modulators of mitochondria, in particular, the ratio of NAD+ to NADH. So these were all, these mechanisms caused increased serum amyloid A in this in vitro model. Then we went on to look at, well, what decreases the level of serum amyloid A in this particular model system. So we did the opposite data mining by searching for those compounds that would decrease the level. And in this case, we came up with, again, a small number of drugs and test agents, but who fell into very specific, a small number of specific classes: APOA1 mimetic, GLP-1 receptor agonists, a particular PDE4 inhibitor, not the entire class, which was interesting, and mifepristone, which is a glucocorticoid antagonist, as well as various multi-kinase inhibitors. so a different set of agents. But what was extremely striking about these results is the fact that all of the regulators that increased serum amyloid A have been associated with increased cardiovascular risk in people, and all of the agents that decreased serum amyloid A in our in vitro model also were associated with decreased risk of cardiovascular events in people as well. And so we got a very tight correlation between the level of expression of this particular protein in this model system with risk for cardiovascular events in patients. And moreover, these results help us connect multiple known risk factors for cardiovascular disease. We know smoking, stress, and inflammation are risk factors, and comorbidities, as well as explaining how drug exposures may impact our risk of cardiovascular events too. So we can separate these mechanisms into those that promote cardiovascular toxicity in patients and those that actually support cardiovascular safety. And moreover, when we put together these mechanisms into a hypothesis for how serum amyloid A is controlling cardiovascular risk within the vascular wall, we have identified or developed a mechanism, tissue mechanism for how this might work, that inflammation and infection lead to increased inflammatory mediators, including IL-6. We get increased serum amyloid A. It has serum amyloid A, one of its main activities is to actually suppress monocyte recruitment and differentiation into foam cells in the vascular wall. So, actually, serum amyloid A promotes the recruitment. It suppresses a suppressive mechanism, so it promotes recruitment and activation and differentiation. These differentiated macrophages produce factors that drive smooth muscle cell hyperplasia and calcification of the vascular wall, which causes hypertension and, again, the increased risk of events, of cardiovascular events due to this biology. So this, and then we can see how serum amyloid A actually is regulated by external mechanisms here. And therefore, by applying this platform, this type of approach with panels of assays, identifying a predictive assay through the use of a data mining a large reference database, we now, at the end of the day, have a much more improved understanding of the disease biology, which helps us, of course, not only identify new drugs or more efficacious drugs by keeping out of this, avoiding this particular biology when needed, but also helps us understand how to manage patients and clinical trials because of the involvement of environmental factors as well as the target of interest for the program. Okay, so now let me tell you about a second case study about thrombosis-related side effects. As you know, thrombosis is the process by which blood coagulates in the vasculature and forms clots. Very normal part of wound healing, but it also occurs in pathologic settings like deep vein thrombosis, stroke, and pulmonary embolism. And it's also a process that's caused by Ebola, which is actually a consumptive coagulopathy because it depletes all of the coagulation enzymes, causing coagulation. Once those enzymes have been depleted, leads to hemorrhage. So it's a very important biological process in both health and disease. So it's associated with exposure to particular drugs as well as environmental stimuli. Exposure to smoking and pollution, particularly through polycyclic aromatic hydrocarbons, which are in pollution and cigarette smoke. Exposure to contraceptives, such as selective estrogen receptor modulators, the SERMs, and various other drugs, mTOR inhibitors, second-generation antipsychotics, and oncology drugs, for example, crizotinib. So it's caused by a number of different drug types and also, as well as environmental exposures. So a few years ago, we had identified, similar to the first case, a biomarker signature in one of these assay systems. In this particular case, it was increased cell surface levels of a protein called tissue factor in a human primary endothelial cell model system of the vasculature. This was identified through a project that we worked on with the Environmental Protection Agency on the ToxCast program, and published a few years ago with Nicole Kleinstroyer and Keith Hauk at the EPA, among several other colleagues there. So tissue factor, that protein, was again quite interesting because it is actually the primary cellular initiator of blood coagulation. So a very suspicious finding is that these some of these drugs and environmental compounds that are known to be associated with thrombosis-related side effects increase the level of this particular protein in an endothelial cell system. So again, we turn to data mining to better understand the regulation, the mechanisms controlling the tissue factor expression in this in vitro system, and doing the same process by taking our signature increased level of tissue factor, comparing it to the reference database, which at the time was smaller, and identifying a number of compounds, but about less than, I think, 1% of the, or 2% of the database, but identifying these compounds actually fell into a number of known target classes, which we were able to place into a biological context. And it turns out that all of these mechanisms are associated with the cellular process of autophagy, which is the way cells eat their own organelles and internal components to generate energy. This is a important process. It has been targeted by oncology programs, neurodegenerative disease programs, but in this case, this is autophagy occurring in vascular endothelial cells. And so we've identified that when endothelial cells are undergoing this process, they increased levels of the vacuolization in cells is causing increased levels, or tightly associated with increased levels of tissue factor on the cell surface. And so, again, this process of doing these testing across a large panel of assays, data mining a large database of of drugs and experimental tool compounds that have known mechanisms, we've been able then here to uncover a much more detailed understanding of the biology that's controlling this particular outcome, thrombosis-related side effects, and connecting mechanisms to each other. So in summary, for these two examples that I've shared with you, certainly the use of these approaches can generate results that are very much more confidently, we can be confident in associating them with the clinical outcomes. Moreover, it gives you a mechanism-based understanding, and anytime there's mechanism understanding, it opens up all kinds of orthogonal assays one can use and generates new hypotheses for either new drug targets or how we are going to use drugs across different patient populations. So, of course, combining these in vitro systems with the large reference database is particularly valuable at identifying previously unappreciated connections that help us understand the disease, but also in a practical way help us be able to design better clinical studies and stratify patients. And keep in mind that also this helps illuminate issues that are going to show up in real world applications, in real world therapeutic treatment of patients, because they have comorbidities. They are taking different medications and are in different environments and under different stressors. And so this gives you a view into the clinical picture at a very early stage. So how might these new approaches be applied or be integrated into our drug discovery paradigm? Of course, rather than only relying on genomics for targets, we make sure that they're either validated in human phenotypic screens or human validated, or even phenotypic assay-derived hits. So just going straight to an assay to identify lead matter. Here, both for targets and phenotypic assays, using structure-based information, one can actually virtually screen millions and billions of molecules virtually, which then can be tested and prioritized in phenotypic assays. Then at the lead op and candidate selection stage, we can apply profiling technologies such as the phenotypic profiling I described to you, but as certainly all of the target-based panels that one can run. There are many panels, kinase panels. There are secondary pharmacology panels. Sarip has many of these running. A number of companies offer access to these panels of assays. And by combining these with the reference databases, we can help identify those assays that are most useful. So again, a role for virtual in silico models. And once we can use these technologies to identify promising leads and lead candidates, leads, certainly, we now need to pay attention to exposure. So, and we know in the exposure field, there's been a lot of work in developing in vitro and in silico tools to predict the absorption, distribution, metabolism, and elimination. And so there's been a lot of work in combining in vitro and in silico approaches here. And I will say for this paradigm I'm showing here, the critical thing about the predictive models in these cases is that they're all local. very focused on a particular activity, mechanism-based, and with a large enough data set to be and sufficiently high performance of these models to be useful. So it's a series of both in silico and in vitro models that would eventually be able to certainly replace animal models. I think we're only now appreciating how poorly animal models are at predicting human exposures and human activities. So the advances in vitro and silico technologies, they don't have to be perfect to be better than what we have today. So if we take all this data, so another aspect of having all of this in vitro and in silico data is the need to have it organized. And I really like a framework such as this for organizing our data and our information in these outcome pathways. These are based a bit on the adverse outcome pathway framework that is being applied for chemical risk assessment in Europe promoted by the and worked on by the OECD, certainly in collaboration here in the United States with the US EPA and NIEHS. This framework allows you to map assays and data onto sort of an organized framework of information that's organized in the hierarchical level, which we know biology is organized. We can use the data to build predictive models, so it's a way to collect data together all in a single place and use those predictive models to support decision making. So this is a framework that would allow us to make better use of all of the in vitro and in silico data and tools that we have by putting them all together into context. So where's the field going? Where are the next important advances happening? Definitely in increasing the biological coverage. There are new cell types, co-cultures, and disease conditions to be evaluated. There's a lot of work in advancing iPSC technology to get additional cell types, to be able to compare patient cells and stem cell technologies. And then, of course, there's the development of more advanced cellular systems, three-dimensional bioprinted tissues, organs on a chip, microphysiological systems. These are still being developed. A lot of R&D. This is a very interesting area, and we've seen some case studies, examples published on the development and use of these systems to make new discoveries, and we look forward to higher throughputs, decreased costs that would enable the ability to build a larger reference database, for example. That remains to be done in the future. And of course, keeping in mind that a drug is only effective if it can get to the target. And so we still are looking at still seeing advances happening in the area of exposure modeling that enable quantitative risk assessment, and risk is for toxicity, but also for predicting efficacy as well. That's a very important area of research that we're looking for advances to help us. So with that, I'd like to conclude. I have a final slide to mention some of my, certainly my team here at Eurofins. It does take a village. And I've mentioned here a few collaborators that we've worked with over the years. And also, I want to highlight some of the organizations and agencies that are promoting non-animal methods and helping to get these technologies developed. So these include government agencies, NIH, FDA, OECD, as well as academic and private organizations. And I encourage you all to support these organizations who are actually helping us take on the reproducibility crisis, or the productivity crisis and the reproducibility crisis, we hope too, but definitely the productivity crisis in drug discovery. So with that, thank you, and I'll turn it back over.
Thank you, Dr. Berg, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask the Question drop-down box located on the far left of your presentation window. Type your questions into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what are the limitations of the cell types used in these methods?
Okay, there are a few limitations. Cost can be a limitation, and that's why scale is important to keeping down costs. There is the donor variability is a limitation, and so one has to incorporate the analysis of donor variation in these model systems. And then availability. There are many cell types that are simply not available. A lot of work going into those: pancreatic beta cells, neurons, oligodendrocytes are some of the cell types that we'd love to have human in vitro cell types models for.
Thank you. Our next question is, how does this work compared to the use of cardiomyocytes for cardiotoxicity?
Great question. Most of the work that people are aware of with cardiomyocytes right now have to do with arrhythmias. And so that is a whole other mechanism of cardiovascular toxicity, which is only explains some of the some of the drugs that have cardiovascular toxicities due to arrhythmia problems. And in that field, it's a little easier because there are good clinical measures or reasonable clinical measures of arrhythmia, unless it, of course, in the case of a new drug for a particular indication, that they had no reason to suspect that it might cause arrhythmia. Causes that. So that's an area where there's a lot of separate research going on, but it explains a particular mechanism of cardiovascular toxicities. And if you notice that there are oncology drugs that have the mechanisms that I talked about today. So it kind of explains why it's been so hard to nail down cardiovascular toxicity mechanisms, because there are many.
Thank you for that answer. Our next question is, when will FDA start accepting non-animal preclinical studies?
So, great question. FDA will accept it now. How FDA works is they want you to show that your molecule is safe. And so everything that you put into a package for an IND, for example, is to answer that question. In many drug classes, like biologics, antibody biologics, for example, may not have a relevant species. And FDA has been open in the past to starting a clinical trial with molecules typically at a very, very low dose, to make, and then do a dose escalation. They are open now, but I would say that it is historical and legacy that is driving the use of animal studies in clinical in drug development, and it is typically just the conservative mindset is that if something goes wrong, I've done what we've always done. I've done my animal studies, so I've covered myself, and I think that is what's happening in drug development. When the data is convincing enough that we can replace animal models, so we need a lot of examples, right? We need a lot of examples where we would have identified problematic compounds or made the same decision as preclinical, then these will be adopted. It will take a lot of data and education.
Thank you. Okay, we have time for one more question. Our last question is, has this approach been successful for other toxicities such as CNS or kidney toxicity?
Great question. We have not done any work in this area. There are a lot of in vitro assays that are being worked on for these tissue types that have shown very interesting results and certainly encouraging preliminary results. But it takes the ability to test a very large reference data set to fully validate an assay for predictivity. And I think that still remains to be done.
I would like to once again thank Dr. Ellen Berg for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.